All right, so welcome everybody. It's my pleasure to introduce today's speaker, Armin Locke. Armin completed his PhD in Cambridge with Wolfram Schultz, looking at the role of midbrain dopamine neurons in economic decision making. And he then went on to do a postdoc with Matteo Carandi, Kenneth Harris, and Adam Capex at UCL and CSHL, looking at the role of frontal dopamine neurons in perceptual and economic decision making. And currently he's a group leader at the Department of Physiology, Anatomy and Genetics at Oxford University, studying the neural mechanisms of learning and decision making. And today Armin will tell us about the dopaminergic and prefrontal basis of learning from sensory confidence and reward value. Armin, the floor is yours.
Thank you so much, Nadine, for introducing me and for inviting me, both Nadine and Steve. Very, very nice. I'm always very humbled when I talk to your crowd, because you guys have done such nice and interesting work conceptually that for us working on animals, it's always like whatever I say is going to be quite obvious. So really bear with me. We spend a lot of time thinking about like viruses, brain signals, and we get less into the concept that your groups and your research have been developing. 80% probably is going to be very obvious to you, but please bear with me. Also, if I start saying things which are not making sense, please do stop me. I have a little baby boy and he's not really letting me sleep, so please just stop me and ask questions. So yeah, better to keep it between the talk rather than at the end of the talk. And yeah, today I'm going to talk about the so just a second, if I can. Okay. All right. So today I'm going to talk about some of the work that I have been doing before in London as part of my postdoc work, as well as some of the more recent work that we have been doing in Oxford. So I will start with previous work and then coming at the end toward the recent work that we are doing in Oxford. And they are at the end a bit less digested, so we are still really working on them. They are on bioRxiv, but we are expanding on experiments and trying to push with other type of experiment. So I'm going to start with a simple scenario of visiting a farmer market, a food market, and thinking about making decisions here. So if you consider yourself in this scenario, in this situation of walking in this market, maybe perhaps not even a very familiar market, a market which is relatively new to you. And if you're trying to make good decisions, it is actually quite a tricky job, because you will need to really process all this incoming sensory information. You need to identify all these fruits and vegetables. You need to identify the colors, evaluate their smell, and at the same time, you need to think about how much you like each of those fruits, each of those vegetables. Are they too expensive, or are they reasonable? Do they have reasonable price, or did you have a lot of this vegetable yesterday, so maybe you don't really want to have one of them again? And making good decision here is really relying on combining all these type of information, both external sensory signal as well as the internal value signals, and they need to come together if you want to make good decisions here. We definitely don't want to go for the food which is very nice but very expensive, or the one that it looks very nice but I don't like it, or other way around. And combination of these two is the whole core of decision making, and in many aspects of our life. This is just one example that I'm bringing forward here. But if you think about the way that we have been approaching this question of how we make decisions in these scenarios, and in particular in the systems side of neuroscience, the situation is that we have two really divided camp of studies. One of them are value-based decision-making tasks or economic tasks, and in that scenario, we train animals to make choices between stimuli which have reward with different values or different probabilities. So the stimuli in those contexts are very explicit. Animals, like in this case, this monkey can really see those fractals, and we have been always giving them these big objects so they can they don't have any difficulty seeing those. But the difficulty that they have here is really to remember which one is more valuable. So that's kind of the core of value-based decision making in the studies that we have been performing with animals and others as well. And the other camp is the sensory decision making, and that's the situation that we challenge animals to detect the stimuli or discriminate the stimuli, and if they are correct in doing so, they will get reward. And the reward in that context is often a constant thing. It's not really changing much. And these two fields are separated really elegant. They are 30, 40, 50 years of literature behind them, both in animal studies as well as human studies. But at the same time, the more they progress, the more segregated they are. And they rely on different behavioral tasks, as we can see here. They have different computational framework. Like in case of the value-based decision making, we can think about the reinforcement learning models, or the work that we know from utility theory, the work that we know from prospect theory as kind of computational and theoretical framework. And in the case of sensory decision, we think about signal detection framework and drift diffusion models and those type of models for thinking about the underlying computation. And of course, these are also coming with studies in different parts of the brain. Different labs have been interested in different regions of the brain, and they have been focusing on these studies in different regions of the brain. And myself, I was doing my PhD in a value-based decision task, the lab, and then I did some work before and after my PhD on sensory decisions. And I thought maybe there are opportunities to bring these two together under one umbrella and try to perform experiments in animals which have elements of each, with the hope that to understand how these two types of information about values as well as the sensory evidence are coming together to make to form decisions and guide learning. So with that, the talk that I will be giving today is having a few sections. I will be starting with behavioral and computational framework for studying decisions which are informed by sensory evidence and reward value. Then I will be showing studies in which we recorded the activity of population activity of neurons in the frontal cortex when animals, in this case mice, were performing this task. Then we will be focusing on the role of VTA dopamine neuron in this task. And at the end, I will be touching on the neuro studies in which we are looking at the detail of the dopaminergic circuit by looking at the projection-specific dopamine signals during this task. So let me start with a very simple scenario to build it up. So let's think about the very simple scenario, a task of that had trials with different reward values. So here we have left and right as choices, and if I choose each of them, I will get reward with different values. And then maybe unknown to me, after some trial, this reward contingency might change. So the way to that this reinforcement learning will tell us to make decision here is to look back at the history of this reward and form an estimation of value of each of those actions, VL and VR here in this case, and then compare those to make choices. So that's kind of very standard value-based decision, or maybe in this case, two-arm bandit task or similar. Now we can think about the situation that we also include an element of sensory evidence and sensory uncertainty. So I'm going to keep the same reward structure that we have. We have these blocks that different actions are having different rewards. And what I'm going to do is to also add some sensory stimuli. In this case, there's this patch of stimulus on the screen. So if I want to get reward in the first trial here, I would need to choose left side. This is basically, I will need to choose the side which the stimulus is telling me to choose. So if I choose right side, I will get no reward. And in the second trial, if there is no stimulus on the monitor, then I can make the choice and I will randomly get reward. And this goes on and on, and the reward contingency is changing. And you can also see that the contrast of this patch of the stimulus is also changing in different trials. And that's giving me the possibility to effectively, systematically manipulate the sensory level of sensory evidence that the subject is experiencing on every trial. So if I want to make good decision here, I should also consider those, on top of those reward values, I also should consider those stimuli. And I can extract an information about those stimuli in terms of the probability that the stimulus is on the left side or on the right side. And I can multiply those in order to compute the expected value of taking the left action and expected value of taking right action. And compare those to make decisions. So one question, of course, one very simple question is like, if we run such tasks in an animal like a rodent or mice, in our case, can they actually make such decisions? Can they make decisions which are informed by both the value of past reward as well as the level of sensory evidence that you're experiencing on that trial? The second which is a slightly more challenging question to ask is how the learning is progressing in this second scenario. So how these trials, how the sequence of trial is helping animal to learn and better perform this task better. So if we think about learning in the first scenario, in the pure value-based decision, we know from reinforcement learning that one way to learn is to effectively compute reward prediction error. That's the comparison of outcome and the value of the choice in the that I have just taken, and then use that difference between outcome and the value of the choice to take it as an error signal and incorporate that error signal in order to make this valuation, these estimates of value better and better over time. And the question is whether this same type of error-driven learning, delta rule, can also be applied in the second scenario, in which on top of value reward differences, we also have these stimuli which are changing in different trials. And of course, the third question is what are the underlying neural circuits for making such decisions and learning from the outcome. And I'm going to mainly focus on two brain regions, the frontal regions in mice, in particular the medial frontal cortex in mice, as well as the dopamine neurons in mice. And there's a very vast literature on the role of this brain regions in decision making, in particular in value-based decision making. And we are going to study how these brain regions would work in this task, which has element of sensory evidence as well as reward value. So to get there, we develop tasks in which we train mice in exact same tasks which I was just describing as this cartoon. So mice are head fixed. They are sitting in front of these screens, and they have a little wheel underneath the forepaw, and they are water deprived, so they are motivated to work for water. And on each trial, there is a stimulus coming on the screen, and that stimulus is telling the animal to turn the wheel and get reward. So in that case, stimulus on the other side, animal needs to turn the wheel other way around. So very simple, you can kind of visual detection task. And then you're changing, as I just mentioned, you're changing the contrast of the stimulus in different trials. And we also have those reward blocks and reward contingency blocks. So unknown to the animal, every 100 or 50 or 200 trial, you're switching this reward contingency. Okay, so before going to the behavior of the animal, let's think about what ideal observer would do in this task. So if you think about ideal observer in this task, I'm not going to go to the detail of ideal observer, but let's just take it as one model which can be considered as ideal observer. This model is having a psychometric function. So on the x-axis we have the contrast of the stimuli, either very high contrast on the left or very high contrast on the right, and the y-axis we have the probability of making the right choice, right word choice. So we have psychometric functions which are shifted toward the side which has larger reward, and that makes sense from reward maximization standpoint. If the model is unsure about the level of sensory evidence, it would bias toward the side which has larger reward. If the model is more sure about those stimuli, then it would just follow the stimuli and not care about the past reward. So that's the effect of past reward for the ideal observer, and that's the effect of past sensory decision. So in this model, is this ideal observer, it would know that these trials are independent from each other, and it would know that if there was a hard trial if the previous trial was a difficult trial or easy trial, meaning that the stimulus was low contrast or high contrast, that has nothing to do with the next trial. So if the model is doing if getting a reward on the right side for a difficult choice, on the next trial, there is no effect on the psychometric function. You can see that if I separate the psychometric curves based on whether the trial was on the right or on the left, whether it was easy or hard, this ideal observer has overlapping psychometric function, which again makes sense because the trials are independent from each other. So that's ideal observer, and that's what we see in mice. You can see that the effect of past reward is exactly what we were predicting. So this time the lines and the dots are coming from our example animal. So past rewards very much similar to ideal observer, but the effect of past sensory difficulty is very different from the ideal observer. So what we are observing is that these animals, if they make a correct choice for a hard trial, for a trial with low sensory evidence, then in the next trial they end up being biased to that side that they just had reward. If they were doing it for an easy trial, so if they have an easy trial and if they get reward, then that effect is much smaller. They don't have that much bias. So it is like I make a decision, I'm not confident about getting reward. I ended up getting reward, and I end up shifting my psychometric function toward that side a lot.
Armin, can I ask you a quick question?
Yes.
So how do you get the past sensory evidence into the ideal observer in the first place?
So the ideal observer, effectively, that I don't. I consider that the model would know that these trials are independent from each other.
I see.
Yeah. But you can do that ideal observer in a variety of ways. I don't think there is one way to do the ideal observer, but I think in majority of them, I would say if the model would know that the trials are independent from each other, then there would be no effect of sensory evidence. Yeah. But in the same model, it would be effect of past reward because it makes sense to consider past reward, but it does not make sense to consider the past sensory evidence. So in a way, it's really an straw man here. It's just to show that these mice are, to some extent, they are maximizing reward because they care about the past reward, but they are also sacrificing reward because while they should not care about past sensory evidence, they end up doing it. So learning here is happening. It's guided by the past sensory confidence and past reward. So I want to open a parenthesis here about this past sensory evidence. This is the effect that we have seen in mice. But we did another study in which we look at a variety of data sets in different animals, including human, in which we did the same analysis on the behavioral data. And in large number of these subjects, that each of them are dots here, we could see some level of this type of behavior. So you can see RAF data here in olfactory task, in auditory task. You can see mouse data here, visual or auditory, some human visual data. And in many of these subjects, you effectively find, although the task and the trials are independent from each other, some level of effect of past sensory evidence on next trial. The last one, this cross modality, is particularly interesting because the animals are randomly doing trials from different modality. So it's like one olfactory, one auditory, and we still end up seeing that effect in those animals, which effectively suggesting that probably the effect that we see is beyond the modality of the stimulus, is about a computation which is not really modality specific. Okay, so going back to closing this parenthesis and going back to the mice data. Now, what we are hoping to do is to think about simple learning model, which can capture these two types of effects. Some of them being kind of optimal past reward, and some of them be suboptimal, which is in this case, this is past sensory evidence. And to do so, I have been building simple reinforcement learning model which bring together these two types of information. So this model, in every trial, it has an estimate from vision, from the sensory system, and that is stimuli which the model is receiving is effectively generating an internal probability that the stimulus is on the left side or on the right side. And the model is multiplying that by the learned value of left action and right action. So if I multiply those two, then I have the expected value of left and right action, these are QL and QR. I can compute the choice by comparing those QR and QL and make a choice. And then I can take the predicted value of the choice, QC, and compare it to the outcome in this model in order to compute the prediction error, which is delta here. And I use that delta in order to update the stored values. So the model has these few parameters and we can fit it on the behavioral data. So those behavioral data out there that we could see before with normal fitting, now I'm going to replace those with those lines which are coming from model fitting. So the model is capturing both the effect of past reward as well as the effect of past sensory difficulty that we saw in this task. The model is also capturing the learning dynamic of animals. So here we are looking at the population of animals, and this one trial one is the onset of block change when the large reward is changing from left side to right side. So animals are getting biased to the other side, and this type of model is capturing that behavior. I'm going to focus on two internal computations in this model. One of them is predicted value, and the other one is prediction error. So if it now plots the predicted value, if we run this model, let's say with the simulation and plot those predicted values, we can see that the predicted value is increasing as a function of the contrast of the stimulus. So I don't care here about the stimulus whether being on the left or on the right. I just care about the absolute value of the stimulus. This is reflecting also the size of reward. So the predicted value is larger for larger reward compared to a small reward, and it is smaller in the error trial compared to correct trials. That's the pattern that we already have seen also in a lot of studies from other groups, in which way effectively think about signatures of decision confidence. That's very similar to this one, modulated by the value of the reward. That's coming from this equation of the predicted value being the probability of choice being correct, that multiplied by the value of that choice. And the prediction error is really the rather opposite image of this, in which the prediction error is scaled by the size of reward. So the dark green is larger than the light green, and it's definitely larger than the red, which is no reward. And also there's this modulation of size of prediction error based on the difficulty of the stimulus that I had in that trial. So now I can think of those prediction error, the modulation, which is depending on the size of reward, as a factor which explains the first behavioral effect, which was the effect of the shift of psychometric function as a function of reward size. And the modulation of prediction error in this other axis, which is the changes of the stimulus difficulty, as how the model is capturing the effect of past sensory difficulty. The model is computing prediction error, which are different depending on how difficult was the stimulus. It would use those prediction error to push the value of left and right, and train those values to be slightly different in subsequent trial, and that is pushing this model or this model fit to capture the effect of past sensory evidence on choices. Okay. So now that I have this very simple internal computations, now the question is, can we understand the neural representation by going back to what I suggested in decoding from frontal cortex and dopamine neurons? And as I mentioned, it's really large literature on these brain regions. The citation here is nowhere. It's not even 1% of citation that I should have in this slide. But the question that we are asking is what is happening specifically in this form of task, which has the both component of economic decision as well as sensory detection. Okay, so in this first set of studies, we use large scale silicon probe to record the activity of many neurons in the medial part of the frontal cortex in mice which perform this task, the activity of neurons here is really diverse. You can find many types of neurons, but one type of pattern of activity that is often coming out in a lot of our recording is that can be seen in this example neuron. So here I have trials on the y-axis and I have time on the x-axis. Trials are sorted based on reaction time of the animal, and they are aligned based on the stimulus onset. And you can see that those dots, those action potentials which appear on every trial, they are nicely aligned to the action onset rather than the stimulus onset in this case. So a lot of neurons that we are seeing at this in these mice, in this type of task, are really driven more by the onset of the action. By action, I mean when the animal is just starting to turn the wheel. So animals head fix is just little wheel movement, and those are the time that we see the large activity in these neurons happening. So for the sake of this talk, I'm going to look at the activity around the time of action. Let's say 200 milliseconds before the action, 200 milliseconds after action. And now we can see that for a lot more neurons, and you can see this line of red effectively shows that a lot of those neurons are active during that time of action, of course, with different latencies. And you can see by eyeballing these two that a lot of these neurons, they don't really care about left action versus right action. They might separate based on this clockwise or counterclockwise movement of the wheel is quite similar. We can look at those activities and we can separate them now based on the stimulus contrast, only for large reward. You can see that as a function of the stimulus contrast, the responses around that time of action are increasing. The responses are also reflecting the value of the reward. So there's this offset for same actions when they are predicting a smaller reward, and they are different in the error trial, much lower in the error trial compared to correct trial. So this pattern is very much similar to the QC, which I showed you in the model, the predicted value of the choice. So these neurons, at a large proportion of them at the time of those actions are encoding the QC term in the model which I presented. Of course, here we are showing this correlation. The question is whether these signals have any causal role in guiding.
Can I just ask a question about that last slide?
Yes.
Is that the contrast of the previous trial or the contrast of the
These are the contrast. So at the moment, I'm ignoring that story of the contrast of previous trial. Is the contrast of the current trial.
Okay.
Yeah. Yeah. So I'm now going to ask these QC signals that we see in MPFC, whether we can actually evaluate whether they have any causal role in driving the behavior. So we are going to take advantage of optogenetic tools. And what we are going to do is a slightly different form of optogenetics. So so there are different ways of activating and inactivating. What we're going to do here is a very nice way of inactivating, is actually to activate the interneurons. So we activate interneurons, in particular in this case, the PV neurons. And people have shown that this is a nice way of rapidly inactivating the population in that brain region. So you will see channel rhodopsin, but what we are doing is actually inactivation because we are activating inhibitory neurons. And we're going to do that in the task which I described with different stimuli and different reward blocks. And the normal behavior, as you remember, was psychometric function, which was slightly shifted toward the side which has larger reward. So before going to the behavioral data, we can try to inactivate our model and see what happens. So we're going to turn the laser on, inactivate it around the time of those actions, and we can simulate that inactivation by lowering the predicted value in the model. So I'm going to inhibit those neurons, lower the predicted value on those neurons because the fire rate was scaled with the QC. So I'm going to lower those predicted value. And the consequence of that is this very short epoch of inactivation is that the prediction error will be larger. So, and that larger prediction error in my model, if I do a simulation, is going to cause a larger shift of psychometric function that you can see on the upper right side. That is the model simulation, and that's exact behavioral result that we see from animals. So we can see that the shift of psychometric function is slightly larger in trials in which we are inactivating at the time between the stimuli and action, the MPFC neurons. So that is a little bit messy. I'm going to compute it a little bit different across animals. So blue is the average of those suppressions, and control is the situation without those inactivation. And you can see a larger change of the shift of psychometric function in that situation. And this is also showing up in those learning curves which I showed you before, like from the onset of the trial of the changes in the block. You can see that there is this appearing to be a larger shift of psychometric function when they're inactivated. So the representation which I showed you in the previous slide, in the neural representation at the level of MPFC, it looks like it has a causal role. if I inactivate those signals, if I inactivate those computational predicted value, the behavioral consequence is that at least the way that I interpret is that the prediction for the animal is lower as a result of that inactivation, and that is causing a larger prediction error. And the behavioral consequence of that larger prediction error is a shift of psychometric function in this behavioral paradigm. And I'd like to take up this as an example of effectively causing learning by changing prediction. So in animal studies, we have a lot of situations that we cause learning by changing the value of outcome. Like you give one reward larger and the animal is learning that side is better. But here we are having an example that we are causing learning by changing prediction. We are manipulating neural activity around the time of a stimulus or between a stimulus and action, and we are causing a shift of psychometric function, which is the hallmark of learning in that scenario. Okay, so
can I ask you a quick question?
Yes.
So this is super cool and incredibly precise. I'm just trying to figure out in my head whether, like, can you differentiate an effect on learning rate? So it just seems like here they're just more reward guided and less stimulus guided. Can you differentiate that from encoding these probabilities or predictions?
Right, I don't think so. I think that this is a great question. So I don't, it's hard to dissociate whether it is about worse perception or whether it's about increase of the impact of past reward, right? Or as you would say, learning rate. In this scenario, I can't dissociate those two. Both of them would give me a shift of psychometric function. There might be a hint on being dissociating those two, because if the perception is really bad, then the slope of psychometric function also changes, right? It would not only stay like this, but that to me is I'm not sure at the level of data it's easy to make those dissociations. So the analysis that we did in terms of a statistic, the slope of psychometric function is not changing in these two scenarios in the population of animals. However, I think it's still tricky, because in the psychometric curve, you might have a range of sensitivity, and you still get more or less quite equal psychometric curve. So that's why I'm staying a little bit unsure about the answer to that question.
Please, while we're stopped, could I ask a question?
Yes, of course, yes.
About the basic model, and this is a rather basic question. Could you define PL and PR? I think you said these were probabilities.
Yeah, so effectively you can consider. Yeah, go ahead.
It would make sense to call them probabilities if you were manipulating the probability of reward on the left or the right. But since it's contrast dependent, I don't think you mean that. On a particular trial, say at a contrast of 0.1, the stimulus is either on the left or on the right, so the probability is 0 or 1.
Right.
So in effect, what you're meaning is that this is an estimate.
Exactly. Yes.
By the animal.
Yes.
So
you can consider
that estimate would have to take account of the strength of the signal, but also, if it's to be converted into a probability, the animal would have to know the distribution of the noise. Is that correct?
Yes, exactly. So you gave the best answer. I wouldn't have given that answer. Yes. So you effectively have an external stimulus, and from that external stimulus, which has a single number, you generate an internal representation, which is a Gaussian distribution with the mean of it around the number that you have from external stimulus. And then that distribution is, then we can cut that distribution to left and right, based on the criterion, and then we end up having the probability that this stimulus is being on the left side or on the right side. So exactly as you described, it's an internal estimate of, by seeing that stimulus briefly, how much I believe that the stimulus is on the left side or on the right side.
Okay, just very quickly, maybe you'll get to this, but do we have any idea where that computation is carried out? Does this fall into your sensory computational box or into your value computational box?
This is again very good question. I think if you ask a visual neuroscientist where that is happening, they would probably say this is happening in visual cortex. But and it's probably some form of it is happening in the visual cortex. If you ask others which are working on other brain regions, other cortical regions, they might say that this is a second order computation based on sensory evidence, which is coming from visual cortex to other brain regions, let's say to parietal cortex or to frontal cortex. And this second order computation is happening in those brain regions. So to me, we are dealing with the hierarchy of computation, which is starting from visual cortex and ending up in higher brain regions. And I think on each of those levels, you might have signature of those computations. The core difference between these two, I think, in terms of separation, is to what extent they are modality independent versus modality specific. We have behavioral studies, and many other groups have behavioral studies that show that probability, that internal probability, could act as a sensory independent or modality independent calculation and influence the behavior that way. And what I showed you in the rat data from cross modality, in which animal is doing different modalities of the task, is also suggesting the same. Nevertheless, I don't think it's easy to exclude the similar computation at the level of visual cortex for modality-specific type of computation. Is that clear, or did I just give you the wrong answer with no?
Yes, that's very, very clear. Thank you.
Okay, thank you. Okay, so I'm going to change the gear a little bit and go to the VTA dopamine activity. We are going to do exact same behavioral experiment. At this time, we're going to image the activity of these neurons by expressing a specific viral construct in these cells, which allows us to measure the activity, the calcium dynamic of these neurons, the population of these neurons during the task. So you can see down there like a raw data, the responses going up and down for a stimuli. And I'm going to make the same analysis as before for this neural data. So one interesting thing here is that the responses at the time of a stimulus is really graded by the contrast of the stimulus, which is making us in neuromodulation feel very, very happy, because it shows like how fast these signals can work in rapid form of perceptual decisions. So as I said, the dopamine activity, we are going to do the same analysis, contrast of the stimulus separate by the size of reward, as well as whether this choice is correct and error. We see very similar thing that we saw in the MPFC, in the frontal cortex activity. Very similar representation of Q value, QC, and those shaded regions are coming from the behavioral data, the behavioral fit. So very similar to MPFC, we see the representation of QC in the activity of this population of neurons around the time of a stimulus, between the stimulus and before the reward comes. So this representation, although we have this representation, but and it's very similar to MPFC, but there's a stark difference between MPFC and dopamine here. And if we do this optogenetic manipulation, which I did in terms of the frontal cortex, in case of dopamine neurons, we do not see any effect. So you recall that for the MPFC, I was stimulating I was inactivating at the time of a stimuli and action. I saw those change of shift of psychometric curve. In case of the dopamine neurons, when we are doing this manipulation, we are not seeing such an effect. So there is a representation in these neurons, but at least with this type of manipulation, we can't change the behavior by changing those neural activity. Okay, so that was the responses at the time of the stimulus in the previous slide. Now I'm going to look at the responses at the time of outcome. So very much what we expect. Responses are larger reward compared to a smaller reward compared to error. And we can again separate those based on what I was doing before, contrast of the stimulus and size of the reward, correct and error. And what we are observing here is that at the time of reward, there is this representation of prediction error, which are not only scaled by the size of reward, but also by the difficulty of the stimulus. So you can see this, let's say the dark green up here. So the size of the reward is the same. There is no stimulus on the monitor anymore, but the responses that we see in these neurons is still reflecting whether the stimulus was difficult, around 0 contrast, or whether the stimulus was easy, around 0.5 contrast. And this is the case of mice, and we previously observed very similar effect, very similar responses when we were when we were analyzing neural data from putative dopamine neurons in primates. Very similar effect of the difficulty of the stimulus on the responses at the time of reward. So it seems that something that is holding beyond rodent, and we can see the same signature in primate literature. Okay, so now that we have this prediction error, the confidence-dependent prediction error, just as a terminology for them, because they are reflecting the sensory confidence. Now what I'm going to ask is whether these signals are actually effective in driving the behavior. So let's focus on just one of those. And what we can do as a toy analysis is to think about those prediction errors for easy stimuli. They are relatively a smaller prediction error. And the effect that they have on the next psychometric curve, because they are a small prediction error, is a small shift of psychometric curve. So the small prediction error is doing a small updating in the next trial, and therefore the shift of psychometric function is small. I can now look at those larger prediction error for the same size of reward again. And here the updating is larger for the next trial, and the shift of psychometric function is larger. So now I'm going to try and see whether we can effectively see this effect by looking at the next trials as a function of size of dopamine in the previous trial. So we look at the activity of dopamine neurons at the time of reward, and we see what happens in the behavior in the next trial. And we're going to separate the trial based on whether the dopamine activity at the time of reward was a small or large, with an arbitrary threshold of 65%. Okay, so what we can see here indeed is the case that even if we control for the size of reward, when the dopamine activity at the time of reward was large in the previous trial, then we see a larger shift of the psychometric function, a larger updating in the behavior of the animal in the next trial compared to previous trial. And this is just to bring it back to the behavior, is just really we believe that this is the basis for this behavior that we are seeing. The prediction error are changing based on the contrast of the stimulus, based on the difficulty of the stimulus at the time of outcome. Those are causing different sizes of dopamine activity, and those dopamine activity are causing different levels of updating, which depends on the contrast of the previous stimulus. And this is causing the shift of psychometric function in a way that depends on the difficulty of the previous stimulus. Okay, so here I'm really trying to get to the situation of like looking at the neural activity and conditioning the behavior based on what was happening in the neural activity in the past trial. So it's a smelling a little bit like having causal effect, but one can say there's still kind of, we are really still looking at the correlation of the neural activity and behavior. So we're going to now test this causality by, again, using optogenetic tools. So this time we are doing two types of manipulation. Either we are stimulating these neurons at the time of outcome, or we are inhibiting these neurons at the time of outcome. And what we could observe are very strong and bidirectional shift of psychometric function in these two scenarios. So in the case of activation, if I have an animal which receives these pulses of dopamine, like let's say performing a right action, let's say, and if the animal is receiving those pulses of dopamine, then the psychometric function is shifted toward that side for those stimulation. And if I change this experiment to be the suppression, so animal is doing the correct action, and rather than receiving rather than having normal activity of dopamine, you have a suppression of activity of dopamine. This is causing the animal to shift away from that action and shift away the psychometric function to the opposite side. I find also this from the standpoint of dopamine stimulation quite interesting, because if you think about very old studies of manipulation of dopamine activity, and there are like classic studies by Old and Milner, you could think that if you have a stimulation of these neurons, the animal works so much to even, they would even let go the reward, and they might even struggle to survive. But if when we think about that, and in this context, what we can see is that the manipulations which are closer to the normal regime of dopamine activity are actually really causing this nice titration between the level of sensory evidence as well as dopamine stimulation. So like in case of activation, you can think of situation that is like when the contrast is very high on this side or that side, animal is still receiving those manipulations, that dopamine activation. But because he's seeing the stimulus nicely, he is not affected by those dopamine activation in the previous trials. So dopamine activation is or suppression on the regime which is similar to the natural activity or endogenous activity of this neuron can work really nicely and can be titrated very nicely with even subtle behavioral stimuli, which are in this case are this patch of stimuli that we have on the screen. Okay, so having said that, I'm going to, if I don't know how I'm doing with time, so I'm gonna see what's happening. Yes, okay, that's looking good with time. I'm going to touch a little bit making the story of dopamine more complicated than what it is. And I'm going to think about projection of specific dopamine signals during this type of behavioral task. So we know from anatomy that these neurons, which are in VTA or substantia nigra, they have dense projection to different regions of the brain. In this case, you can see like very strong projection to different regions of a striatum, ventral striatum, dorsal striatum. The consensus about the kind of the anatomy is that some of these neurons, let's say those which are in lateral substantia nigra and substantia nigra, they have more projection to dorsal regions of a striatum. And the ones which are in VTA, they have more projection to ventral regions of the striatum. So there is a rather partial segregation of these neurons projection to different regions of the striatum based on where they are originating. And there are nice studies in simple situation in which they recorded the activity of these neurons. By activity of these neurons, I mean the activity of these axons in different regions of a striatum. So the same imaging which I did on cell body, you can also do on axons, because those axons are also expressing those viral things that we are using for imaging. And this study is effectively showing that if we image from very dorsal region of a striatum, in the second row, we can see some responses at the time that animal is locomoting. So the second column is locomotion. There's really no behavioral task here. It's like animal is locomoting and sometimes it's getting reward. So no context, it's just very passive situation. So you can see this dorsal region's locomotion signal, but then when the animal is receiving reward, there is not much signal. And then if you go back down into ventral striatum and record the axons of dopamine neuron in the ventral striatum, you see much smaller modulation by locomotion and much stronger signals at the time of reward. So that's already telling us that there's probably something going on if you think about our task in terms of these different axons which are coming to different regions of a striatum. We did exact same experiment, and this is the work that Morgan and Peter in my lab have been doing. And it's still evolving, but we have the first set of results now on bioRxiv. And so we have the preparation to have this expression of this this particular GCAM in the axons, either in the dorsal regions or in the ventral regions, and we have a fiber which is either going deep into the striatum or shallow in the striatum. So we record those axons during the behavioral task which I was talking about. And if we do so, we find striking differences between the ventral striatum and dorsal striatum at the time of a stimuli. So ventral striatum is quite similar regardless of whether the stimulus is on the left or on the right. I'm separating here based on ipsilateral and contralateral. They are very much like VTA. They scale the responses based on contrast. But in the case of dorsal striatum, the story is a little bit different. They only care about the contralateral stimulus, but not the ipsilateral stimulus. We can look at the responses, again, at the time of a stimuli, separate based on size of upcoming reward. In case of ventral striatum, again, we see something similar to VTA. There's differentiation prior to outcome. but it's not the case for dorsal striatum. And we can separate based on whether this choice is going to be correct and error. We see very similar again to VTA in case of ventral striatum, but different in the case of dorsal striatum. So if I want to summarize what I have here, in that upper plot in the ventral striatum, you can see that responses are modulated by the contrast of the stimulus. They don't care if the stimulus is on the left or on the right. As long as the stimulus is predicting reward, they are firing those axons. And they reflect the size of reward, and they are different in the error and correct triad. But unlike that, in the dorsal striatum, we see very strong, rather exclusive stimulation, the activity of these axons only for the contralateral stimuli. So up here, ventral striatum, very similar to VTA, and the model which I showed you can account for this. This can be really explained by prediction error computation under sensory uncertainty. And in this case, we have this strong encoding of contralateral stimuli. It's not that easy to explain this with the model that I presented. The model is really caring about whether the stimulus is rewarding or not. It doesn't care about left or right. So that's the case for the stimulus. And the next slide, I'm going to show you that these dorsal axons are also caring about contralateral actions. So up here, you can see the activity. So they are aligned to the time of action. They are already separated before the action because you remember that they are coding the stimulus contrast. So that is just the remaining of the stimulus coding. At the time of action, they also have these signals which are going up only for the contralateral action. So the axons not only doing contralateral stimulus, but they are also responding to contralateral actions. They don't care much about, at the time of action, they don't care much about the size of reward, and they have some separation of correct and error for at the time of action. So if I want to put these two action responses and stimulus responses together in a cartoon, we effectively have this scenario that for the dorsal striatum, there is strong contralateral stimulus responses, relatively insensitive to the size of reward or the accuracy of the choice. And there is a second bump in the activity at the time of actions, and they appear for correct choices, but not for error choices. We are really thinking about what this signal are doing and how they would be useful for guiding this behavior. But you can also think about also in the context of making association between the stimuli and action. Maybe these signals actually can be useful. So they are really, what they care about is that this was a contralateral stimulus, this was a contralateral action. Maybe they are really one of the ingredients on making simple association between a stimuli and an action. And then if we look into like other papers which are thinking about the plasticity of corticostriatal connections, we can see signature of we can see things which are in favor of this idea. So in this work, which I have in this blue box, there are mice which are learning from scratch that there is a stimulus and they need to make a lateralized action. And you can see in the second plot, you can consider the y-axis is complicated here, but you can consider that as the level of the efficacy or plasticity of corticostriatal synapse. So how strong that synapses are. And what we can see in that study is that as the animal is learning to make this contralateral action in response to stimuli, then this level of corticostriatal plasticity is increasing. And so this study is not considering dopamine at all. And I think what we have in our study is really the ingredient which is adding to this plasticity story, because corticostriatal plasticity is something which is driven by dopamine. So you have cortical projection, striatal activity, and what is causing this plasticity is the release of dopamine in that site. And what we are suggesting, and we are still working on it, is that these lateralized signals that we are seeing in the dorsal region for dopamine axon is perhaps useful for making this simple form of association. And with this, I would like to slowly summarize what I have shown you. I showed you the behavioral paradigm that we have in which these animals, in this case mice, are continuously learning from past reward and past sensory confidence. I showed you that a simple reinforcement learning model that includes an estimate of sensory confidence can account for this form of learning. And I focus on two internal variables in this model, the predicted values and prediction errors. I showed that MPFC neurons are integrating sensory confidence and reward value and encode the predicted value. And those predicted value signals at the time of stimuli and action do have a causal role in learning. In case of VTA dopamine neurons, their prediction errors are reflecting confidence in getting the reward. And the responses are accounting for how the value of reward and how the sensory confidence can work together to shape future decisions. And I showed you some of our recent work on projection-specific signals. In case of ventral striatum dopamine axons, the signals are very much similar to VTA. They encode confidence-dependent prediction error. But in case of dopamine projection to dorsal striatum, they are mainly concerned about the contralateral stimuli and actions. With this, I would like to acknowledge past and present collaborators, people in the lab, as well as founders of the lab. And thank you so much for your listening and for questions. And if there are other questions, I'm very happy to take them now.
Great, thank you. Let's all unmute and give Armin a big round of applause. Awesome. Steve, maybe you can stop the recording.